Evidence for relationships between organisms. Genetic comparisons using DNA and proteins. So how can comparisons of base sequences in DNA be used to see how closely related organisms are? What is DNA hybridization? How can we use comparisons of amino acid sequences in proteins? How are immunological comparisons used? Genetic comparisons using DNA and proteins. Classification systems were originally based on observable features, but now we can use a range of evidence to determine evolutionary relationships. When organisms evolved, visible internal and external features and their molecules change. DNA determines the proteins, including enzymes and proteins, determine features. Changes in features can arise due to changes in DNA. Comparing DNA and proteins of species helps scientists determine the evolutionary relationships. DNA comparisons. Comparison of DNA-based sequences. When one species gives rise to another in evolution, DNA of a new species is initially similar to the species that gave rise to it. Due to mutations, the sequence of bases in new species of DNA will change. Over time, new species accumulate more differences in its DNA. Species which are more closely related show more similarities in their DNA than distantly related species. As millions of base sequences in every organism, the DNA contains much information about the evolutionary history. DNA hybridization. When DNA is heated, the double strands separate into two complementary strands. When it is cooled, the complementary bases on each strand recombine to reform the original double strand. DNA hybridization is used to compare the DNA of two species by the DNA from the two species is extracted, purified, and cut into pieces. DNA from one species is labelled using radioactive markers. Then it is mixed with unlabelled DNA from the other species. The mixture of both sets of DNA is heated to separate their strands. The mixture is then cooled to allow the strands to combine with strands with complementary base sequences. Some double strands that reform are made up of one strand from each species. New strands called hybrid strands are identified because they are 50% labelled. The hybrid strands are then separated out and the temperature is increased in stages. At each temperature, the degree to which the two strands are still linked is measured. If two species are closely related, they will share many complementary bases. Therefore, there's more hydrogen bonds, so stronger the strand is. The stronger the hybrid strand, the higher the temperature that is needed to separate it. The higher the temperature, the higher the hybrid strand splits, the more closely the two species related are. The lower the temperature at which it splits, the more distantly related. So basically, because if you have two similar species, they will have more similar complementary bases, so therefore there'll be more hydrogen bonds, therefore it'll take a higher temperature to split them. If they're distantly related, they'd hardly have any base pairs which match, therefore it won't take a high temperature because there won't be very many hydrogen bonds linking them together. DNA-based sequencing and classifying plants. Until recently, the classification of flowering plants was based on appearance, so the plant was placed into two groups: monocotyledons, which are single seed leaves, and dicotyledon, which is two seed leaves. They have been reclassified by DNA sequencing of 3 genes which are found in all plants, and a phylogenetic tree for families of flowering plants is devised using DNA sequences. Protein comparisons. Comparisons of amino acid sequences in proteins. The sequence of amino acids is determined by DNA. The degree of similarity in amino acid sequence of a protein in two species will reflect how closely related the two species are. Once the amino acid sequence for a protein is determined, the sequences can be compared. You can count the number of similarities in each sequence. Immunological comparisons. Antibodies of a species respond to antigens on proteins. The albumin in blood serum of another. The serum albumin, or antigen, from species A can be injected into species B. Therefore, species B will produce antibodies that are complementary to the antigens on the albumin from species A are produced. So, the serum extracted from species B, this contains antibodies specific to the albumin antigens from species A. The serum from species B can be mixed with serum from blood of species C. Antibodies respond to antigens of albumin on species C, and the response is formation of a precipitate. The greater number of similar antigens, the more precipitate is formed, and the more closely related the species are. The fewer the number of similar antigens present, the less precipitate, the more distantly related the species are. Okay, so you can see that human and chimpanzee have the same amino acids at different positions. The gorilla has a few differences at position 23 and 104. The orangutan, compared to the human, it's only got one that matches.